All right, it's time to start. Can everyone hear me all right? If you can, raise your thumb and nod your head or what have you. The link will be placed on Blackboard. Again, I know this is not ideal. I'm very sorry. I don't know what I did this morning, but I was bent over in the shower to clean the rubbish from the drain. And I stood up and I turned, and then this wave of pain shot through my lower back. And I said, oh, ow, that hurt. Oh, it'll go away. No, it didn't. And now walking is quite painful. So hopefully I'll be able at least to make an appearance for lab tomorrow. This doesn't feel like it's going to go away quickly, but I hope it does because, man, it hurts. I've not really had a back injury like this. I played football in high school. I had a helmet in the back once. It didn't hurt as much as this. So getting old sucks. That's kind of the whole theme here. So thank you for being flexible. I appreciate it. I know it's not ideal, but I'll see everyone in lab tomorrow for the enzyme kinetics lab. Tomorrow, it's going to be very important. Somebody has a computer tomorrow and that you're graphing your results while you're doing the experiment. Unless you properly graph the data, you won't know if you have a good run of the enzyme. And if you don't, you have to repeat it. But just looking at the data, you won't be able to tell unless you graph it. So while people are doing the experiment, somebody should be on the computer graphing it so that you can see if on the Lineweaver-Burke plot, the double reciprocal plot, you have a straight line. If you don't have a straight line, then you need to do it again. That's kind of the long and the short of it. So if you don't need to do it again, great, but you don't know unless you graph it. So you need to graph while you work. Whistle while you work, graph while you work, that sort of thing. All right, let's have a word of prayer. I don't mind if you guys have your cameras on. I do ask that you mute your microphone unless you want to ask a question. Asking a question is perfectly fine. You can use the chat function at the bottom. Do you see where it says chat? There, I just sent a chat to everyone. You all see that? So you can send a chat back, you know, you're ugly, you know, whatever, something like that. And then it should give me a prompt that I'm seeing it. So, and if I continue on and don't see it, just unmute yourself and say, hey, you, I have a question. All right, let's have a word of prayer, then let's begin. Father in heaven, thank you for this day. Thank you, it's a nice day. We pray the lab tomorrow goes well, that students will get good graphs, and that we'll be able to get through it expeditiously. Thank you for our TA who does good preparation. We pray the derosinase works well and that people would get a reasonable graph. Now, Lord, help us learn. And Lord, please heal me because I'm not of much use to anybody as an invalid. In Jesus' name we pray, amen. Very good. Let's now take a look at the PowerPoint. The subject for today is protein structure. In particular, we will take a look at the different types of secondary structures. One of you in your feedback session said you wish we could talk more about secondary structures. That's exactly what we're going to do. One other person asked, what's the largest protein discovered thus far? The largest protein discovered thus far is probably titin, which is in muscle. Is there a way to have students research something and then explain their results to the class. If there's time. We have a lot to get through. We don't necessarily have time. It looks like we sped through some things. Yeah, we did. We have to. That's the nature of it. And yeah, those are mainly the questions. The speed was the concern. So yeah, it's the nature of the beast. Okay, so a conformation. That is the 3D arrangement of the constituents of the protein, and that 3D structure is called the protein conformation. If that conformation is a particular 3D arrangement that allows the protein to perform its function, then it's called the native conformation. The native conformation. Is protein conformation static? The answer to that is no. Proteins very clearly are moving. They're constantly breathing and moving around. There's also thermal movement that's associated with the protein. Are there limits to the dynamicity of protein conformation? Yes. If you denature a protein, it no longer works. So there's a limit as to which, inside of which protein doesn't have any function anymore. So protein structure is not static. Protein structure is relatively dynamic. Some protein structures are more dynamic than others, as we'll see. Are there limits to the dynamicity? Yeah, there are. Because the protein needs to have a certain structure in order to be able to perform its function. What molds the protein into its conformation? The local structures a protein forms will then mold it into its 3D conformation, because there's only so many ways to fit those local structures into a coherent function. And the biggest force molding a protein into its native structure are hydrophobic interactions, because the protein is going to fold around a hydrophobic core, and then the hydrophilic bits will be on the outside to associate and interact with the aqueous environment. So hydrophobic interactions drive protein folding. So the conformation the protein assumes, when it's ready to do what it does, when it's ready to execute its function, that's the native conformation. That conformation is maintained by interactions that are relatively weak, at least by themselves. Collectively, those interactions are actually stronger. So collectively, they're relatively strong. When you add them up, additively, they're strong. By themselves, they're relatively weak. So the tendency for a protein to maintain that native conformation, or even to come into and form that native conformation, is referred to as its stability. The stability. Since weak interactions help a protein maintain its stability, the amount of energy that separates a native conformation from a non-native conformation is somewhat small. It's about 20 to 65 kilojoules per mole. And proteins that assume their native conformation will tend to stay that way on the average. Proteins that don't fold properly don't tend to unfold on their own and refold. They have to be unfolded and then allowed to refold, usually with help. The cell has proteins that help folding proteins get it right, and then help improperly folded proteins try again. They're called chaperones and chaperonins. We'll talk more about those toward the end of the lecture. Disulfide bonds can greatly support a native protein structure. Disulfide bonds tend to be found in proteins that are either outside the cell or they're found inside organelles. So the interior proteins usually are quite hydrophobic. If you look at the 3D structure of sequences and look at where the hydrophobic stretches are, and then take a look at where those stretches are in the folded protein, you'll find they're typically in the interior of the protein. Now we can show you this actually in a very simple, well, relatively straightforward way. So we're going to stop sharing. And we're going to share. We're going to share this screen here. And we're going to go to PubMed. And now we're going to go to all right, so next thing we're going to do is we're going to go to the oh boy. Okay. Let's do this. NCBI. And we're going to go to structure. So proteins. Then we're going to go to protein structure. Let's see here. Where's the structure? Oh, here it is. Here it is. That's one. Okay. So this is the viewer. And if you want then to view this, their structure. So let's take a look at hexokinase. Okay. Here's a crystal structure of human hexokinase. And we're going to download that. Whoops. Had that on the wrong thing. That's what we wanted. They keep changing this, and there we go. Okay, so here's our protein. You can grab on it and drag it around, and you should have the sequence of this protein here. And so you can, okay, come on, load, buddy. So if you were to take, say, Um, okay, this piece here that's been highlighted over here. And then you want the, they used to have the sequence right in front of you. They changed that. So this is one of the hexokinase molecules here. So we want to select okay, so side chains. Oh boy, they really changed this. That used to all be in the drop-down box. Not anymore. Okay, let's do oh, there we go. There it is. There it is. Okay. All right. So here's our sequence. Now, if we take a look at this sequence, we can see here, what you can do is you can highlight bits of this sequence here, like that. And if you do that, it'll show you where it is on the protein. So that's been highlighted here. You notice how this loop over here, and then if you highlight, say, this here, it'll give you this loop here. Now, if you look at this here, notice that it's proline and valine, isoleucine, glycine, so on and so forth. Notice how that's interior to the protein. If we keep looking, we look for some sort of hydrophobic stretch here. And if we look for one, cysteine, there's one, not a very long one. Okay, here's one. Yeah, here's one right here. Okay, there. See how that's interior? That's an interior piece. And you can go on and on and on and on with these sorts of kind of interior sorts of pieces here. And notice, once again, we have an interior piece of the protein, like that, there. All right, I just wanted to show you that because that essentially can be used to identify clumps of proteins and stretches of hydrophobic protein. They change that, but that it takes a little bit of a learning curve with that. It's CNR when you go to the NCBI website, and that shows you essentially bits of protein. As you look at the sequence, essentially, you can highlight bits. It'll show you where in the crystal structure it falls out. So you have sizable chunks of your protein that are hydrophobic that are inside the protein structure itself. And those hydrophobic stretches usually associate first. They do so to escape the water. And then when they escape the water, the polar amino acids then will start to interact with the water on the outside. So the interactions between the polar amino acids and the water can potentially be destabilizing. That's simply because an interaction with the solvent means that you have interactions within the protein that aren't occurring. And this is what we mean here. Every hydrogen bond made within the protein, and that stabilizes the protein. If you have a hydrogen bond made between the solvent and the protein, that means you have a protein-protein interaction that is not made. So that's potentially destabilizing. That's why big pieces of protein that flap outside the protein can be potentially destabilizing because they're not giving you interactions within the protein strand itself. Now, peptide bonds have a planar character to them because of the carbonyl. So that bond between the carbonyl carbon and the nitrogen, there's going to be some sharing of the electrons between that bond. That means that the bond between the nitrogen and the carbonyl carbon will be shorter than what you might have predicted because of resonance structure. So there's going to be a partial negative charge on the carbonyl oxygen, partial positive charge on the carbonyl nitrogen attached to the carbonyl carbon. So if we look at the bond like this is very electronegative. You'll have some sharing of this double bond here. That means you have a dipole that's been set up here. That gives you opportunity for hydrogen bonding with the backbone.
Dr. Bradovich, your
Yeah.
slides aren't moving.
Slides aren't moving, okay? Let's try it again. Screen to, okay. Okay, now they're moving? Yes? Do they move now?
Yes, they are.
Yep,
Okay, good.
they're moving.
All right, good. Okay, let's go back to, okay. Right, so this dipole that you get here between the carbonyl carbon, the carbonyl oxygen, and that nitrogen there, you're going to have some bond sharing. And that means that gives you the opportunity for proper hydrogen bonding here. So the peptide bond and the group on either side of it is called a peptide group. Peptide group, and here's an example of a peptide group. Here's your peptide bond, and this one, that constitutes a peptide group. That means if we take our pen here, that means this right here, that would be another peptide group. And then over here, there would be another peptide group, and so on and so forth. So that carbonyl carbon, that's sp2 hybridized, it's planar. So that means your peptide bond has a planar geometry about it. So when you think of the background, the backbone of the protein, think of small little planes that are next to each other. And then the amide group will be in trans to one and then in trans to the other. So here it goes down and then up and then down and then up. Same thing with the R groups. The R groups will go up and then down and then up and then down. Whole thing will be in a zigzag. Carbonyl carbons will go up and then down, up and then down. If this is up, the hydrogen here on the amino group is down. If the carbonyl is up, the R group is up, albeit it's jutting out to the other side. And then so on and so forth, repeating over and over and over again. So because of that double bond, you have a C to N bond that's not able to rotate. But the other bonds can rotate. So if we look here at our carbonyl-carbon, this bond here doesn't rotate. This bond does, and this bond does. Now, because these two can rotate, there are certain angles that are allowed, there are certain angles that aren't. If you take a look at this steric hindrance here, that tells you right away there's a certain angle for this bond that's not allowed. So these angles, they're called phi and psi. Phi and psi. The value of these two angles will depend upon the R group. Some R groups allow very wide rotation. Like, for example, glycine allows very wide rotation of phi and psi. Tryptophan does not. Proline really doesn't. So it's going to depend on the identity of that R group. So your peptide backbone, you can think of it as a series of planes that rotate around this common carbon, that alpha carbon, and then rotation around the bonds to that alpha carbon, bond the alpha carbon to the next nitrogen, the alpha carbon to the previous carbonyl carbon. Those are the bonds of phi and psi, and some of those angles are allowed, some of those aren't.
Do those does it maintain that conformation once it starts to, like for the alpha helices? Do you still have those planes that are preserved?
Yeah, they are. And the bond angles are going to be locked into certain angles that are different than what's normally allowed by having those because of the stabilization provided by the backbone that gives you that alpha helix or beta sheet or whatever. That will lock them. But the little planes, that will still be maintained. So between the C alpha and the C bond here, that's phi, psi, and between the C alpha and then the next amine, that's phi. And rotation around those is in a 0 to 360. And so each amino acid has a preferred range for phi and psi. And if you look at this, trying to use the 3D ChemDraw here to show you essentially the rotation around those bond angles. Here, you can already see this rotation causes some problems right here. And this is obviously an alanine and what have you. And here's a phenylalanine here, and blah, 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 so on and so forth. So this shows you essentially those two bond angles. So those amino acids with smaller R groups have less restricted phi and psi values relative to those that are large. They'll be more restrictive. So if we're asked the question, which has a wider range of phi and psi, glycine or tryptophan, I would hope you would in unison answer, go ahead and answer.
Glycine.
Glycine, thank you. And then your book essentially shows you the ranges of phi and psi that you find in proteins. So within the blue here, those are what you see in proteins. There's kind of an isolated little island out here, isolated one down here. This over here is pretty much forbidden territory. It's just sterically not allowed. And this is called a Ramachandran plot. A Ramachandran plot. And there are certain secondary structures that map to certain areas of the Ramachandran plot, as we'll see in just a bit. Okay, secondary structures. So secondary structures, if the primary structure, let's see, let me, so let me pick on somebody. Oh, Crystal's, your picture comes up first for some reason. So Crystal, unmute yourself. Tell me what the primary structure of the protein is.
It's the order of the amino acids.
That's exactly right. Exactly right. You can mute yourself now. Yeah, that's right. Exactly right. Amino acid sequence. That is the, yeah, okay, you can react to that if you want. That is the primary structure. Secondary structure are local conformations assumed by stretches of the polypeptide chain. So if this is a polypeptide chain here, and it assumes a local structure. So let's just do a little local structure here. So that's an example. That's an example of a secondary structure. There's still the rest of the protein, but this local structure here is a secondary structure. And then the tertiary structure will involve taking these secondary structures and fitting them together in a coherent 3D structure. So that's a secondary structure. Do you think you understand now what a secondary structure is? So there are two most common types of secondary structures, alpha helices and beta sheets. Alpha helices and beta sheets. Let's talk about alpha helices first. So to illustrate an alpha helix, I use my thought toy, and that is a slinky. Because an alpha helix is a lot like a slinky. So we have here a coil, essentially. And this coil starts here and coils down this way. You'll see this one is underneath this one, and it stretches out like that. And that is a very good example of an alpha helix. So that's essentially my example of an alpha helix. So the alpha helix is the simplest conformation a localized group of amino acids can form. It looks like a corkscrew. It looks like a slinky. It's stabilized by the backbone, not by the R groups, by the backbone. The R groups can contribute to stability, or they can mess with the stability of an alpha helix, but they're not involved with the formation of an alpha helix. So here's a picture stolen from your book, and it shows you those associations, hydrogen bonds between elements of the alpha helix. So you have a carbonyl oxygen, hydrogen bonding with an amino hydrogen. You have an amino hydrogen, hydrogen bonding with a with a carbonyl oxygen, and so on and so forth. You have a nitrogen, hydrogen bonding with an oxygen, and so on and so forth. Are any of these bonds by themselves strong? No, but collectively they're strong. And when they form, they can lock the protein, that portion of the protein, into a corkscrew-like structure called an alpha helix.
So is the reason why the whole protein is not an alpha helix, is it just because of the destabilization of the R groups? I mean,
Some stretches of protein more readily form alpha helices than others. Those stretches that readily form them will usually form them first, and then the protein will fold around those stretches, and then other stretches that do other things. Some stretches of the protein don't readily form alpha helices. Some do, as we will see. And they will essentially help with the folding of the protein. They will essentially nucleate the folding of the rest of the protein. So your carbonyl carbon or carbonyl oxygen will hydrogen bond with the amide group above it in the helix. Where you have a turn, you're going to have a turn about every 3 or so amino acids. When you get below there, you have this bit above this bit. You now have an opportunity for a hydrogen bond. And then you have an opportunity for a hydrogen bond. Then the opportunity for a hydrogen bond. And then hydrogen bond, hydrogen bond, hydrogen bond, and so on and so forth. And your peptide bonds in the backbone of the alpha helix participate in this and form and stabilize the helix. This makes this a very thermodynamically stable structure. And you can see essentially the structure looking down on it. This structure was first proposed by Linus Pauling. He was apparently in bed with a cold, and he started drawing amino acids on paper and cutting them out and doing paper models with them. And when he did a paper model, he got a corkscrew, and he won the Nobel Prize actually for that thing he did in his bed, cutting out, essentially doing cutout dolls almost, making amino acids and cutting them out and so on and so forth. So you look down on it, you can see there's like a tunnel in the middle of it, just like here in the slide. And then if you look at it straight on, it looks kind of like a tower. And it's a pretty tight structure. This is not a picture from your book, but this shows you very nicely the hydrogen bonding in the backbone. Also, the R groups stick out from the backbone. So they're going to stick out this direction and maybe interact with the R group below it. So the stability of the alpha helix depends very much upon the amino acid chain that forms it. They will either strengthen it or they will destabilize it. And Irma, I don't know if I got back to your original question, but it really depends upon the primary structure. Typically, bulky amino acids will interfere with the formation of alpha helices. Also, proline and glycine are inimical to the formation of an alpha helix. If you have a proline in an alpha helix, you usually have an alpha helix that has a kink in it. Can anybody guess why proline and glycine might restrict the formation of an alpha helix? Anybody?
Is it because of how far they can turn the rotation? Glycine can turn more and proline can turn less, can rotate.
Yeah, that's pretty much it. So the glycine is too flexible. So the helix will undergo certain amounts of thermal breathing. So it'll, with the glycine, you've got, it's kind of like stacking your books on top of a really small box. You stack your books on a really small box, and then the box is too small. The books then just go tumble right to the ground. That's kind of what a glycine does in a helix. It causes it to kind of fall apart. Proline has the opposite problem. Proline is too rigid, and proline is at such an angle that it tends to kink the helix. There are helices with kinks in them, so things can fit over them, can form, and then this can fit over it. Those aren't very common, but they do happen. Proline is pretty alpha helix unfriendly. All right, so proline doesn't allow the rotation around this end to alpha carbon bond. Also, that nitrogen atom doesn't have any substituent for proper hydrogen bonding. That makes for even bigger issues. Glycine has just way, way, way too much flexibility. So the twist of your alpha helix guarantees that you have some sort of interaction between an amino acid above and amino acid below. That amino acid above and below will be anywhere from 3 to 4 amino acids apart from each other. Therefore, the identity of the amino acid at the beginning and the amino acid at the end are pretty important for proper alpha helical stability. The amino terminus usually is going to have a negatively charged amino acid, usually, or maybe there'll be one near the amino terminus. The carboxy terminus, it's not unusual for it to have a positively charged amino acid. That gives the amino acid, the alpha helix, a dipole. And that dipole will further stabilize the alpha helix. Now, some amino acids show a stated preference for being in alpha helices. And they're listed here in both single letter and triple letter code rather. And so if you see these stretches of amino acids, there's a good chance that they might be in an alpha helix. What is a beta sheet? That's a second type of secondary structure. So beta sheet is a kind of zigzag structure rather than a helix. So you got this zigzag kind of structure, and this zigzag structure usually has one stacked on top of the other, and that's called a beta sheet, sometimes called a beta pleated sheet. A beta sheet or a beta pleated sheet. And instead of a helix, you get this zigzag sort of structure. The polypeptides line up one on top of the other to form a beta sheet. So in your beta sheets, your hydrogen bonds are formed between the peptide backbones of the adjacent polypeptide chains, just like what you see here. Once again, it's the backbones that will stabilize this structure, not the R groups. The R groups will protrude in an alternative fashion, so they'll be down, up, down, and so on and so forth. Beta sheets can be parallel or they can be anti-parallel. So anti-parallel, this one goes in this direction. It's going from front to back. This one's going from front to back. So this one's going in this direction. This one's going in this direction. This is anti-parallel, anti-parallel. But if we switch it around, now in this case, because of the kink over here, this goes from front to back, and this goes from front to back. So this is parallel. And this is shown in the next slide. So anti-parallel, you can see these are heading in different directions. Front to back, to front to back, to front to back. Anti-parallel are definitely the more common of the beta sheets. Here's parallel. This goes from front to back, front to back, front to back. This is parallel. This is not as common as anti-parallel, but you still see it. I want you to see here, notice where the R groups go. They're grouped together like this. All right. Because of this arrangement, beta sheets do a better job of accommodating bulkier amino acids than do alpha helices. So here's some examples of anti-parallel beta sheets and then parallel beta sheets. And notice how they're packaged together. Can alpha helices and beta sheets be packaged together in a protein? Yes. And those secondary structural motifs, alpha helices and beta sheets, Those structures can be then grouped into other secondary structures, sometimes called super secondary structures. Super secondary structures. And they include things like the beta alpha beta loop. We have two beta sheets, and between them is an alpha helix. So this is a beta alpha beta loop. That's an example of a super secondary structure. The other is an alpha alpha corner. Alpha alpha corner. You have two alpha helices, and those two alpha helices have a stretch between them that's not structured, and it allows them essentially to be at right angles. That's an alpha alpha corner. That's an example of a super secondary structure. We'll get into that turn structure, what that turn structure might look like. There are restrictions for amino acids that can participate in beta sheets. So less bulky amino acids form them quite well. So for example, beta carotenes have a very high alanine glycine content, and silk fibroine is very rich in beta structure.
So are beta sheets better or worse at accommodating bulky amino acids than alpha?
The betas will accommodate them better. In terms of forming them, it takes them a little while to form them, but they definitely accommodate them better. Alpha helices will accommodate them if they're at the end. Then you have one at the end, it's not a big deal. You have one in the middle, doesn't work very well. So here is a structure of silk fibroine, and you can see the glycine, alanine, serine, glycine, alanine, blah, 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 so on and so forth. And it repeats over and over and over again. It's very, very rich in beta sheet structure. And here's a cartoon, essentially, of that structure, very rich in beta sheet. And it forms, essentially, the long threads of a spider's web being mixed with silk fibroine and so on and so forth.
So, so in this structure, would this be considered parallel or anti-parallel?
That structure, silk fibroine is anti-parallel. That's because the protein essentially loops out and comes back, loops out and comes back. You get the picture.
So basically, if the C terminus that's on top lines up with the amino terminus at the bottom, that would be considered. Is that so? If the amino terminus ended up on the other side, that would be parallel.
So the amino terminus would be over here. And the C terminus would come out over here. And as you can see, it's traveling from front I take my arrow here. Let's see. It's going from front to back, 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 to back, and so on and so forth. Sorry, my fault. So you can see here, that's why it's anti-parallel. The strands run in opposing directions. Does that help? Here's what the spider's web looks like once it comes out and dries and folds, and that's what it looks like. So here are some amino acids and their tendency to be in one or the other. Some of these have a high tendency to be in both. Some of these have a high tendency to not be in either one, but to be in a turn. Some of them have high tendency to be in both, so they're in both. So it really varies. And you'll see the glycine and proline aren't in much of a mood to be another one, but they like to be in turns. Same thing with asparagine, histidine, so on and so forth. Okay, let me pause here. I'm gonna stop sharing. Any questions? Anyone? All right. Question. Slides are moving now. Okay. There we go. Okay. Now your globular proteins will fit the beta sheet and the alpha helix together, and that'll form your tertiary structure of the protein. And in between your beta sheets, alpha helices are turns, where the polypeptide will change direction. And these turns are called beta turns. And in the beta turns, you have either glycine or proline. And the glycine should be pretty easy to ascertain. It has tremendous flexibility, and that provides essentially the ability essentially to turn. Prolines are also common. Now, proline exists in a trans or cis isomer. And almost all prolines exist in the trans conformation. Now, I tell you, if you wanted proline in a turn, would it do better in a turn in the trans or the cis conformation?
The cis.
The cis, because it's turning. So in fact, 6% of all proline residues are in the cis conformation. And there is an enzyme in the ER lumen called proline protein isomerase, PPI, proline protein isomerase, and it converts transproline to cisproline so that cisproline can participate in beta turns. So cis prolines are usually formed in beta turns. And the beta turns are usually going to be at the surface of the protein where hydrogen bonding interactions can occur. So here are some different types of beta turns, and it looks to me like we're out of time. So I will see you guys in lab tomorrow. If I end up doing a lot of sitting tomorrow because my back is not cooperating, you'll know why. I'm going to ice it and put hot heat on it tonight with stretching, lots and lots of stretching, and then sleep on it. We'll see how it is tomorrow. Don't forget, make sure you read through the lab. Make sure you know how to do the Lineweaver-Burke plot. And I would set up an Excel sheet so that you can go from spreadsheet to Lineweaver-Burke plot. For tomorrow, that'll save you time. Okay, that is all for today. I will hang out here if you have any questions. In the meantime, have a great rest of the day. So long.
Hope your back gets to feeling better.
Thank you, Irma. Makes two of us.
Have a good night.
Thank you. You too.
So, lab tomorrow, as I was